Macintosh. Sorry. I too would like to thank Drs. Bonewald and Fielding for their hard work organizing this meeting. 48 hours ago, I had no idea of the diversity and complexity of regulatory interactions that existed between skeletal muscle and bone, and actually going home excited about some new experiments. So for me, that's a good meeting. So thank you for putting it together for us. What I'd like to present to you today is some information concerning another way that bone can communicate with skeletal muscle, and that's via bone-marrow-derived cells that can exit the bone marrow and can contribute to muscle regeneration following injury and disease through several mechanisms. What I'd like to do is give you an overview of the type of cells that can contribute, bone-marrow-derived cells that can contribute to muscle regeneration, and then provide some evidence about the different mechanisms through which they can act. So that bone marrow-derived cells can contribute to tissue regeneration is well established. It's been known for well over a decade that bone marrow cells could have this function. We know from extensive literature that bone marrow-derived cells can contribute to the regeneration of liver, brain, cardiac muscle. We heard some great examples this morning from Drs. Rubin and O'Keefe how bone marrow-derived cells could contribute to healing of bone. And also, where there's good, strong evidence that bone marrow-derived cells can contribute to regeneration of injured and diseased skeletal muscle. There are a wide variety of bone marrow-derived stem cells that can contribute to regeneration of these different tissues. Amongst this group that's listed here, the first two, hematopoietic stem cells and side population cells, or SP cells, are best known for being able to contribute to the muscle compartment and participate potentially in regeneration. So hematopoietic stem cells are CD45, CD34 positive cells that are lineage negative, meaning that they're at a very early stage of differentiation. SP cells are identified primarily not by the phenotypic markers that they express, but rather by their ability to extrude Hoech's dye via specific transport mechanism. It's actually likely that SP cells are a subpopulation of hematopoietic stem cells, since it's been shown that they express many of the same phenotypic markers and possess many of the same functional characteristics. So hematopoietic stem cells, early in their differentiation, when they're multipotential cells, exist in the bone marrow, and they begin to undergo differentiation while in the bone marrow. While in the bone marrow, they begin to enter lineages of cells that will eventually can lead to the production of fully differentiated cells of every blood cell type. Much of that differentiation, though, occurs not within the bone marrow, which has the appropriate niche for development of these cells, differentiation of these cells into blood cells, but some of these cells, when they're still multipotent, will exit the bone marrow where they're exposed to other niches that can affect their differentiation and potentially guide them along other routes of development. If we look at more clearly at some of the developmental pathways that are available to hematopoietic stem cells, we can see that in the bone marrow primarily are the multipotential hematopoietic stem cells. The first developmental decision that they make is whether to enter the common lymphoid progenitor path of differentiation or the common myeloid progenitor. The common lymphoid progenitors give rise to all the lymphoid populations, T cells, B cells, and also natural killer cells, whereas cells that differentiate from the common myeloid progenitor form all other non-lymphoid blood cells, including red cells and all other white cells, such as neutrophils, monocytes, and macrophages. So a question then is, at what stages of differentiation along the hematopoietic cell differentiation pathways are these cells able to be diverted from the hematopoietic lineage to enter the muscle lineage, where they have the potential to fuse with muscle cells and to contribute to regenerative pathways? So one of the many papers, and this is a paper that I think is especially wonderful, it's from Margaret Goodell's lab years ago, where she tested whether or not hematopoietic stem cells had the potential of contributing to muscle regeneration following muscle injury. What she did is she first isolated She first isolated the SP population of cells from the bone marrow, and you can see they're very low prevalence cells, less than 0.005% of the total bone marrow population. And she then characterized those and showed that they express the stem cell antigen 1. So these are SP cells that are of the hematopoietic lineage. Then she took a single hematopoietic stem cell. This is from a mouse that expressed a fluorescent reporter. She took a single fluorescent hematopoietic stem cell and transplanted it into a recipient mice that had been irradiated. She then gave time, several months, for that single cell to repopulate the hematopoietic compartment of the recipient mouse. Then she injured the muscle and looked to see if any of the cells derived from the daughter cells of that single transplanted cell had joined the myogenic compartment. And what she found is that the daughter cells, or cells derived from the daughters of that cell, were able, in the injury context, contribute to the myonuclei and muscle fibers. So what you can see in A here, this is a stained section where you can see the fluorescent outline of a muscle fiber. What you can see in B is these are red nuclei, so they're expressing the reporter, indicating that they are derived from the daughter cells of that original single transplanted cell. And then in D, you can see the merged image. So it's possible for the hematopoietic stem cells, so this is definitively a hematopoietic stem cell, that in the injury context can fuse with muscle fibers and potentially contribute to the regenerative process. So the next big question, though, is what are the stages of the hematopoietic cell differentiation in which cells have this capability to have this plasticity to exit the hematopoietic lineage and then enter the myogenic lineage? So what we know from Goodell's work is that it could be any stage downstream of the multi-potential hematopoietic stem cell. So in trying to dissect out which of these many different stages of differentiation cells have this plasticity, in a study by Helen Blau and her colleagues, she used a similar muscle injury model, but what she did is she sorted cells according to different phenotypic markers indicating that they were at different stages of differentiation along the hematopoietic stem cell differentiation pathway. What she found, if we start at D prime at the bottom here, you can see that this is a cell, or this is a muscle, injured muscle, in which there have been transplanted multipotent hematopoietic stem cells. These are expressing a green fluorescent reporter, so you can see that these undifferentiated cells, not only hematopoietic cells, not only are they fusing with the injured muscle, but they're also transcriptionally active. So you can see the green fluorescent protein distributed throughout the cytosol of the muscle fibers. Blau also looked at later stages of differentiation. So she sorted out a population of cells that were at the common myeloid progenitor population, shown in C prime here. And what you can see is there was also successful engraftment and fusion of cells at that stage of differentiation. But if she transplanted cells later in differentiation, for example, when they had entered mature myeloid lineage cells, such as shown in A prime or B prime, she observed no cases in which the transplanted cells were able to fuse and join the muscle compartment. So what this tells us then is that at early stages, when we have completely multipotent hematopoietic stem cells, they are able to fuse and be redirected into the muscle compartment, but also cells that once they've entered the myeloid lineage, they still retain that capability for a period of time of being redirected to become muscle cells rather than myeloid cells. But big question, how important is this? This shows that it can happen, that fusion can happen, but how large is the contribution of these cells to the muscle compartment? Well, data from the same paper by Margaret Goodell addressed that question, and what you can see here is the percent donor-derived myonuclei in injured muscles from the 3 mice that had the highest levels of engraftment. And what you can see, my cursor isn't working here, so I hope you can see under the injured column that in each of these animals in which there were the 3 highest levels of engraftment, the percent of myonuclei that were derived from that transplanted cell was less than 1/10 of 1%. So this is feasible for redirection to occur from the hematopoietic lineage into the muscle lineage. And this could be important for therapeutic use, but it leaves us wondering how important is this in regular muscle adaptation, this plasticity of hematopoietic cells. What she also observed is that when she looked at non-injured muscle, she never saw any cases of engraftment of the donor nuclei. So this tells us two things. It's an uncommon event, and it's also an event, this fusion of the hematopoietic stem cells with muscle fibers. It's an event that occurs only in the context of muscle injury. Okay, so we know that this can happen, fusion of these early, relatively undifferentiated cells, the hematopoietic and myeloid lineage with muscle fibers can occur in an injury context, but on a very, very low frequency basis. What we also know, though, is that injured muscle and regenerating muscle, that there are tremendously elevated numbers of myeloid cells in the tissue, although these cells are incapable of fusing and contributing through regeneration through donating myonuclei to the injured muscle. What we find is that mature myeloid cells, in particular macrophages, are the most vastly predominant phenotype of myeloid cells in regenerative muscle, and this is independent of the model of injury or adaptation, whether it's toxin injection. We've heard some studies reported here about cardiotoxin injection muscle, whether it's freezing, crush injury, but also in modified muscle use. What I'd like to show you a bit is some data from the hind limb suspension reloading model that Dr. Bloomfield presented earlier today. This is the rodent model, remove weight bearing from the hind limbs. Within 10 days of removal of weight bearing, muscles like the soleus will atrophy by 40%. Then return the animals to loading. They see that as increased loading of the muscle. It causes damage to the muscle, inflammation, repair, regeneration, and growth. So it's a very structured path of injury, inflammation, regeneration, and growth. What we find in this model is that rapidly after reloading, we see large numbers of mature macrophages. So these are expressing F480 antigen, which is present on the surface of fully differentiated macrophages. We find them scattered throughout the muscle tissue. Although these are all fully differentiated macrophages expressing F480, they are not all of the same phenotype, though. If we look at an inflamed lesion in muscle, this is actually not from hind limb reloaded muscle after hind limb suspension, this is from a dystrophic muscle. But if we look at an inflammatory lesion in muscle where we have damaged muscle fibers and we stain with fluorescent antibodies to different phenotypic markers, what we'll see is this sort of complex mixture of macrophages that are expressing different levels of different phenotypic markers that reflects the complexity of the phenotype of the macrophages that are present in these inflammatory lesions. So this isn't a monolithic population of macrophages, even though they're all terminally differentiated mature macrophages. What's determining the phenotype of these macrophages is the cytokine environment in which they differentiate. So pro-inflammatory cytokines like interferon gamma and tumor necrosis factor will activate macrophages to an M1 phenotype, which are pro-inflammatory macrophages. Anti-inflammatory Th2 cytokines, such as IL-4, 10, and 13, will activate macrophages to an M2 phenotype, which is an anti-inflammatory phenotype. If we look at the time course of the appearance of these different subpopulations of macrophages over the course of muscle reloading and compare it to what's occurring in the muscle relative to injury and repair, what we see is that soon after reloading of the muscle, first of all, this blue line, this is an indicator of muscle damage as measured by membrane lesions, indicated by the influx of extracellular marker dye through lesions in the membrane. We see that at about 1 day to 2 days of reloading, we're reaching peaks of muscle membrane damage, and this coincides with peak populations of neutrophils and M1 macrophages that express high levels of the marker CD68. Then, as membranes repair, and as muscle regeneration and growth begins, we see that M1 populations decline and M2 populations increase. So these M2 macrophages are positive for CD163 and CD206. So what we know then is that Pro-inflammatory cytokines will activate macrophages to the M1 phenotype. These are pro-inflammatory macrophages that express high levels of CD68, and they're elevated in muscle at the peak of muscle damage at about 1 to 2 days reloading. We then find that they're replaced by a population of M2 macrophages that can be activated by Th2 cytokines. They're CD163, CD206 positive cells, and they have characteristics of serving anti-inflammatory functions in tissues. Because of the temporal association between the invasion of the muscle by M2 macrophages and the onset of membrane repair, muscle regeneration, and muscle growth, we wanted to test the possibility that the M2 macrophages could actually be driving the regenerative process and increasing growth. So our first approach at this was to deplete macrophages from the regenerated muscle, so doing antibody depletions at this period between 2 and 4 days of reloading. We found that the depletion protocol that we developed was successful. If we look at 2 days of reloading, what we see is there's no difference in the concentration of macrophages in non-treated or treated mice. This is all in soleus muscle. But what we found is doing the depletion between 2 and 4 days that control macrophage populations remained high, so these would be predominantly M2 macrophages, but in the macrophage depleted, we essentially eliminated all the muscles that would have been present in the muscle during this stage of reloading. So what's the impact on muscle injury? Well, what we find during reloaded muscle, using this fluorescent fiber assay, where this indicates an injured fiber, that between 2 days of reloading and 4 days of reloading, that normally membrane repair occurs. And we drop down to essentially 0 injured fibers by 4 days of reloading. However, if we deplete the M2 enriched macrophage population, we see that there's no membrane repair that's occurring between 2 and 4 days of reloading, indicating that the M2 cells are important in driving the repair process in these muscle fibers. So what are the effects on regeneration? So yesterday Brad Olwin explained that a morphological indicator of muscle fiber regeneration is the presence of central nuclei in the muscle fiber. What we see is between 2 and 4 days of reloading in soleus muscle fibers that there's a big increase in central nucleation, indicating a progression of regeneration. However, that regeneration is greatly reduced in the muscles of animals that have been subjected to depletion of macrophages during the peak of M2 macrophage invasion. Perhaps more importantly, what we found is that M2 macrophages are playing an important role in driving the growth of muscle fibers during this period of increased loading. What you can see here is that reduction in fiber cross-section area in the black control animals during the 10 days of muscle unloading, and then between the onset of reloading, 0 hours of reload, 0 hours of reload, and 4 days of reloading, you see this continuous increase in the cross-sectional area of the fibers, indicating that they're growing in response to increased load. When we deplete macrophages between 2 and 4 days of reloading, we prevent any growth that occurs during that period. So this is telling us that the M2 macrophages are playing an important role in promoting both regeneration and growth following periods of muscle disuse. So what could be keying this switch between the M1 macrophage phenotype and the M2 macrophage phenotype? We know that there are a population of Th2 cytokines that are capable of activating the M2 phenotype. So we wanted to know if IL-4, 13, or 10 were serving to provide the switch to turn on the M2 macrophage phenotype and activate muscle growth and regeneration. We looked to see which of these cytokines was elevated during the activation of the M2 phenotype and found no change in expression of IL-4 or IL-13. But what we did find is that IL-10 expression increased significantly in the 4-day reloaded, began at 1-day reloaded, and then further increased at 4-day reloaded muscle. We saw that this increase in IL-10 coincided with increases in the level of expression of CD163, a marker of the M2 phenotype, in 4-day reloaded. And we also found another important marker of excuse me, the M2 macrophage phenotype, arginase 1 was greatly elevated at 4 days of reloading. And there was also a coinciding increase in the levels of expression of IL-10 receptor subunit 1 at the 4-day reloading. So this suggested then that maybe IL-10 was driving the M2 macrophage phenotype switch and enabling them to promote muscle growth and regeneration. So we tested that by mutating IL-10 and seeing the effects of the IL-10 mutation on the macrophage phenotype switch during muscle reloading. And what we found, well, first of all, is that when we ablate expression of IL-10, that the increase in CD163 expression that normally occurs at 4 days of reloading was prevented. Likewise, we found that the increase in arginase 1 that normally occurred at 4 days of reloading, another important indicator of M2 phenotype, was also ablated by null mutation of IL-10. So does knockout of IL-10 have similar effects on muscle repair, regeneration, and growth as depletion of the M2 macrophage population? Well, the results are actually fairly quantitatively similar, where fiber injury using the fluorescent fiber assay, we see no injured fibers in the unloaded muscle. At 1 day of reloading, we see an increase in both the wild type and the IL-10 null mutants, an increase in fiber injury. And in the wild type, we see a reduction of fiber injury between 1 day and 4 days of reloading, but that reduction doesn't occur in the absence of IL-10. Similarly, central nucleation is somewhat attenuated, an indicator of regeneration is somewhat attenuated in the IL-10 null mutants. And then, again, maybe even more importantly, the effects on growth, where we see similar rates of atrophy or identical rates of atrophy during unloading for the wild type in the IL-10 knockout. And then between the resuming reloading and 4 days of reloading, we see this continual growth in fiber size in the wild type that's completely prevented by null mutation of IL-10. So this suggests that the IL-10 switch to the M2 macrophage is a key factor in driving, in at least this model, muscle repair and regeneration. So something that we had noticed about our data concerning the switch in macrophage phenotype following modified muscle loading or any model of injury is that these switches in macrophage phenotypes seem to correspond temporally with changes in patterns of expression of muscle-specific transcription factors that are necessary for muscle differentiation. So that correlation led us to begin to inquire whether or not this switch in macrophage phenotype might also be functionally related to changes in levels of expression of different muscle-specific transcription factors. So we assayed that first in vivo by looking at whether the change or whether ablation of IL-10 expression affected patterns of expression of Myeloid or myogenin, and we found that the number of Myeloid positive satellite cells in reloaded skeletal muscle was not affected by the IL-10 mutation. However, we found a large effect on levels of myogenin expression at 1 day of reloading that were caused by the null mutation of IL-10, where we saw significantly and greatly increased levels of myogenin positive satellite cells in the muscles of the IL-10 null mutants. There are several possible explanations why we could see this elevation. We don't have any data to favor one explanation over the other, although our working hypothesis at the moment is that we're wondering if IL-10 mediated signaling via M2 macrophages is necessary for satellite cells to progress from the stage where they're expressing high levels of myogenin to progress to later stages of differentiation. So we think maybe these satellite cells are getting hung up at the stage of differentiation, which is contributing to their elevated numbers. We don't know that for a fact, though. So we tried to get at this more mechanistically by looking in vitro at relationships between M1 and M2 macrophages and muscle cell proliferation or differentiation. What we did is we isolated macrophages and activated them to an M2 phenotype with IL-10, and were able to show that they were activated by seeing increased concentrations in CD163 expression. Or we activated macrophages to an M1 phenotype using interferon gamma stimulation and confirmed their activation to the M1 phenotype by their elevated expression of iNOS. We then co-cultured the M1 macrophages or the M2 macrophages with satellite cells to look at the effects on proliferation, And what we saw is that M1 macrophages greatly suppress proliferation of satellite cells in vitro, while M2 macrophages had significant effects on increasing rates of proliferation of satellite cells in vitro. So we think that maybe part of the mechanism through which M2 macrophages are promoting muscle regeneration and repair is by increasing the cycling time of satellite cells to accelerate repair. We also looked to see whether these co-cultures would yield any changes in levels of expression of Myo-D and myogenin, and inconsistent with our in vivo data, where we saw in particular with myogenin big effects of perturbing IL-10 expression on levels of myogenin expression, we found that in co-cultures of M1 macrophages or M2 macrophages with satellite cells, we've found no effect on levels of expression of Myo-D or myogenin. It's always hard to explain negative data. It may be that we haven't got the dosing right, the time right. We're still working on this. Or another explanation that seems to me maybe even more likely is that the in vivo environment is going to be so much more complex. There are going to be so many other cytokines there that could be modulating the effects of IL-10 that it makes it hard to see these treatment effects in vitro. We're still working on that, though. So where does this leave us? Well, we think that there is good evidence that bone marrow-derived cells can contribute to muscle, either by showing plasticity in their pathway of differentiation, so in particular cells in the multipotent hematopoietic stem cell population, or early myeloid precursor populations that have the capability of fusing and contributing their nuclei to injured muscle. What we don't know, though, is how important that is in normal adaptation of muscle. What we do know is that as the myeloid cells become more differentiated and enter terminal differentiation, they lose the capability to fuse with injured muscle cells, but instead, as they differentiate into an M2 phenotype, they're able, presumably by soluble factors that we're seeking now, are able to affect both the proliferation of satellite cells and likely their differentiation and possibly growth of fully differentiated muscle fibers. So I'd like to mention the people that did the work that I've shown here for you, in particular Michelle Whaling-Henricks, Armando Villada. So Michelle's a former graduate student, Armando's a former graduate student, Bo Dang's a former graduate student. I'm looking for graduate students. All three of them contribute major parts of the work I just showed you. Thanks.
I have a little time for some questions. Anybody? If not, oh, here's a question. Microphone 2.
Thank you very much, Karasik, Boston. Could you please explain whether M1 is participating in muscle fibrosis?
Yep. Whether M1 participates in muscle fibrosis. There is no evidence that M1s participate in fibrosis. The macrophage phenotype that's most clearly associated with fibrotic processes are the M2, specifically M2A phenotype. So the M2As express arginase 1. So arginase 1, it metabolizes arginine and it drives the production of proline and polyamines. The polyamines will increase fibroblast proliferation, and then the prolines used for collagen production. So if you have prolonged periods of M2A macrophage elevation, that will increase tissue fibrosis.
Thank you.
Is that related to these rare diseases of fibrotic changes in the peritoneum and in the thyroid? And
well, I don't know about the peritoneum. I know that in chronic muscle diseases that it's associated with persistent elevation of M2A macrophage populations. And some of the work that we've done with in dystrophic muscle, where over long periods of time you get an accumulation of connective tissue, when we've knocked out expression of arginase 2 in M2 macrophages, we've greatly reduced tissue fibrosis.
Very interesting. Well, it's time now to...
I had a question.
Oh, I'm so sorry.
That's okay. Jim, do you have any idea of the numbers of nuclei of M2 macrophages versus the number of nuclei in the muscle? So Are they similar to satellite cell numbers? Are they much greater when they're there at their peak numbers, or do you have any idea of that?
When they're at their peak levels, so between 2 and 4 days of reloading, or if it were MDX mice during at 4 weeks of age, where you get peak levels of inflammation, I would say that it's well over 2 orders of magnitude more nuclei that are in myeloid cells than in muscle fibers. You get, so in this reloading model, which is a relatively mild injury model, you will get a few thousand myeloid cells per cubic millimeter of muscle, and in MDX you can get in excess of 100,000 myeloid cells per cubic millimeter. So that just many, many times more than satellite cells.
I'd like to ask my co-chair, Vince Cayoso, to pose the questions that he formulated for this session, and then we'll move on to the panel discussion. Vince, please come up.